first speaker is Gaetan Klein. He is a French psychotherapist specialized in trauma therapy with 11 years of experience. He had an early interest in addictions and trauma therapy and helped over 2,500 clients and trained therapists in this area before joining the Institute 6 years ago. He now focuses on performing and spreading the word about the clinic processes such as TBI, Asperger syndrome and OCD. Formerly suffering from Asperger syndrome, he participates in the development of the Institute's clinic and research. So Gaetan, over to you.
Perfect. So this is an introduction to what we're doing and what we're going to present through the whole symposium. And I wanted to give that presentation because last year, for the previous symposium and symposium before that, some people were a little bit confused about what are we even talking about. And sometimes the fundamentals seem so obvious to us, researchers and therapists, that we forget to teach the basic things. So in this presentation, I'm going to teach you the basic things of what we do. And let's start with the beginning. So how did it all start? Well, our research institute really started in the context of psychotherapy when we were trying to use different techniques to help people ease their suffering. And one of the techniques that we've been developing was using regression to treat psychological traumas in particular. So what is regression? Regression is a principle that if you have an emotional trigger that happens in a situation today, it's generally because your memory has recorded another emotion in the same kind of context in your life before. So for example, if I have a car accident and I am anxious about it when I see a car after that, whereas the anxiety I get when I see a car is not because the car, it's because the accident. So once you have a trauma, it impacts your memory and you have emotional triggering after that. So regression is based on the idea of going back earlier in time when the first trauma occurred so that you can efficiently remove the emotional triggering and the pain associated to it. So by doing regression more and more to these traumatic memories with the techniques that the team developed, which is called whole-hearted healing, we started to see that we could remember pre-birth developmental stages and memories of things that happened before we were born. And we can explore these memories from within as if we were there. And that's where really we started to explore this area because it's really fascinating and it's really key for the science that we're doing now. So it's really surprising for some people, but the body actually keeps a memory of its own development from the smallest, earliest spark of life when we really begin to exist to how we grow through this developmental sequence. And then when we get born, there's so many things that happen already is that we get born with not a blank slate when we get born. We already have a lot of memories from our development. And where it's getting really interesting is that we can use it to explore the entirety of our biology. So let me explain. When we go further in the past, when we explore things that happened before we're born, the further we go, the smaller and smaller it gets until we become very, very small, right? So think about events like cell division or conception. And before that, the egg and the sperm themselves have a history, a memory. So the more we regress, the further we go in the past, and the more these events are biological events. And we call them developmental events because very often there are important moments where our biology change. And that's what we study. And these very early events very often contain traumas. So that's why trauma healing techniques apply to exploring this time zone. And when we heal this early developmental event, it has a profound effect, not only on our psychological well-being, but also it has biological effects. It changes our biology, right? Because if we change things that happened to the first cells that became you, it obviously has effects on your whole biological construction. So one of the key element that we use all the time is trauma, as I mentioned already. So I guess most people understand what stands behind the word trauma. So a trauma is a psychological shock where you have an emotion that comes up, you have a memory in your mind that stays stuck in your mind, and you experience suffering from that memory. And in different contexts that will trigger this memory, the same emotion will come up again and again and again. And these traumas also contains physical damages when we regress on them. And when we were studying the biology of trauma, we found that these traumas are stored in structures that we call ribosomes, which are stuck on a string of messenger RNA inside the cell. So I'm not getting into the complexity of it because there's going to be another talk about that in details. But what I just want you to take with you as an idea is that trauma is equal to epigenetic damage. And epigenetic damage is when our body cannot use its potential. It cannot use the potential of its gene, and we have something that is stuck, that is blocked inside of us. And this is really biological, it's physical. It's not just something that happens in our head because we have a bad memory. It's something that really gets imprinted in our body and at the deepest level, in the cellular level. So that's why trauma healing is very important when we study this, because that's how we can change the biology. When we heal trauma, the epigenetic damages change. And basically, that's what we do all the time. Since we observe that healing traumas has a repairing effect on epigenetic damages. And when these epigenetic damages are repaired, then there is an effect that is passed on the whole body and then to organs, right? So we really start fixing something at the genes level, and then it repercutes, it's carried on, to the cellular level and then to the whole body level. Hopefully that makes sense. If you have questions on that, let me know after. But you understand the implication of that. It's really important because it means that healing psychological traumas actually can impact our physical health and also diseases, as we're going to see. But now you wonder, how can we find the relevant epigenetic damages in order to repair them, in order to have an impact on specific targeted biological mechanisms or diseases? Well, we found out after years of research that there is a key to understanding how this works. And we finally realized that there is a cell that we call the primary cell. And we call it this way because this cell leads all the other, all the other cells in our body. That means that what happens inside of this cell will determine what happens in all other cells. And this cell is really unique, and it contains all the potential for all of our biology. That means our genes, everything that happens in other cells happens in that cell too. And something that is really fascinating is that it doesn't just contain genetic material, but it also contains our consciousness or our being. And so understanding what happens inside this primary cell allows us to link biology and psychology together. Now, each structure inside the cell has a psychological function, and that's why, as you can see in this diagram, it's really important to map out and understand what are these different parts of the cell and what are the psychological functions. And also, it allows us to check if they are functioning correctly or not, because it's not always functioning correctly. Unfortunately, it's very common that there are dysfunctions in various parts of the cell. And these dysfunctions have a negative impact both physically and psychologically. And that also means that if we can find the source of these problems, then we can study psychological and physical troubles and wonder what is the counterpart of this problem inside the cell. If there's something that happens in our body, we can look inside the cell. If there's something that happens in our mind, we can also look inside the cell. Make sense? So to conclude on that, what we do is called subcellular psychobiology, and it's the science that studies the body consciousness link, or the body-mind link, and how it functions inside the primary cell itself, because the primary cell is the first matrix, the first layer where everything is coming together. So it's really a revolution, I think, because this allows us to do things that haven't been done before. For example, we know that all efficient trauma therapies exploit a mechanism inside the primary cell. And this is specifically a branch of biology called epigenetic. I know that my colleague Céline Guérin will give you a full talk on this. And also, we can study other phenomena. For example, there's a lot of people who report out of the ordinary or spiritual experiences. And it's easy to dismiss them and think that it's just an illusion or it's just people's beliefs. But what if these experiences actually have a relationship with biological processes and some things that happens inside the primary cell? It turns out that it's the case. And so these discoveries establish this new science, which is really mixing biology and psychology together. So we call it psychobiology, and specifically subcellular psychobiology. And if you think about it, if the first cell that will become you has a problem, well, then you exist with the problem. There is no other way to compensate for it, because there's nowhere else that you can that your health is coming from. Your health is coming from these very first cells that become you. And so if there are problems in development, if there are damages to the sperm or the egg, then it has an impact on how you grow, how you become yourself. So here are the axioms, the fundamental principles of subcellular psychobiology. First, it has to be compatible with existing science, right? And science starts with observation. So rule number one, all valid observations within psychology and biology must be compatible. From this understanding, it's not possible that two sciences which are true contradict themselves, right? So if there are contradictions, it's important to understand why and to find out where are the theoretical flaws of it, so we can reconcile those two parts. And psychology and biology should not be separated apart science, but they should actually come together. So all valid observation within those fields should be compatible. Also, all theoretical models should be compatible, because we're talking about human beings. Psychology and biology are about life, so they should be compatible. And these models that integrate the two must integrate all the observable phenomena and give them an explanation, right? And including phenomena that previously were disregarded as anomalies. For example, psychologists might not understand biology, and biologists might not understand psychology, and sometimes have a hard time explaining what happens in the other science. So we must have a model that integrates all of this together. And so that's what we're intending to do. With subcellular psychobiology, we are creating a unified model that integrates all of the data from both psychology and biology. And if that model is correct, then it should be able to predict new observations. And that's how you test for a theoretical model. You test the validity by having a hypothesis, running tests, and creating experiences, using logical deduction, and all the other tools from the scientific method. And if it works, then you can derive concrete applications. You must have something concrete that comes out of it. So this is really what we're intending to do. We're intending to bring psychology and biology together, and also to expand on this with this new model. Because science progresses by widening, and new theories integrate and broaden previous models. A famous example is Newton's gravitation. Newton's gravitation is true, but it's true in a certain frame. And if you go outside of that frame, it's not true anymore, but it doesn't mean that this theory is false or wrong. It just means it works in a certain frame. And Einstein general relativity expands this frame a lot. So it includes Newton gravitation, but it also expands on it. And just like if we have a new theory, a new model, it should expand on what's already understood. And that's why psychobiology not only encompasses psychology and biology, but also spiritual experiences. And we come back to this later on. So how does it work? Well, it works just like any other science. So we start with an observation, we have a lot of data, a lot of things we can observe, and we try to make sense of it. So we create a theory. And now that we have a theory, we want to know if that theory is valid, it's true. So we create hypotheses and experiences, and we observe the results. Are the results saying that the hypothesis was right or wrong, or do we need more to conclude? And from all these experiences, we refine the theory. And hopefully, if the theory is valid, then we have applications, and it's an ongoing cycle. So how do we use this primary cell model, this subcellular psychobiology model, to create new applications? Well, you're going to hear all about it through the whole weekend. But one of the things that we can immediately find solutions on are trauma healing, because that's where we started from. So there are dozens, if not thousands, of psychotherapy techniques all around the world, and most of them work on trauma. So if they do work on trauma, since we know that trauma are biologically embedded inside the cell, then we can wonder, okay, how does this psychotherapy technique work at the cellular level? How does this one work? How does this one work? And we find what are the different mechanisms that they exploit, and then we can use it to improve this and find the ones that work faster, better, are more reliable on the long term, etc., etc. And we can do this because we can explore what's happening inside the cell. And the exploration of epigenetic allowed us to identify 4 types of trauma. And these 4 types of trauma, they require different techniques. And if you use one technique that works only on one type of trauma, you won't have an impact on the other 3 types. So by knowing what we have to deal with, we can adjust our techniques. So this messenger RNA string, we have a lot of them. Average people have thousands of these inside our cells. And that means that we have a potential for trauma, because each string equals a potential trauma. It might not be a trauma that manifested in your life so far, but it's a potential. It's like a pre-sensitivity to certain situations. And they also block metabolic pathways. So since we have a lot of them, it also means that our biological power or capacity is not fully expressed. And that's really one of the most fascinating things of our work, is to explore this unexplored potential. Now, let's look at some applications, and you will hear details about this in the next presentation by Dr. Mary Pellicer. But one of them is also related to trauma healing. So we start with an observation. The body actually blocks trauma healing in some cases. It does if the trauma is linked to survival. It's something that we have observed again and again and again. And it makes trauma therapy stars at an average of 70–80% from my observation, which is great. But why does the body block trauma healing, and how can we find a way around this problem? Now, by observing what happens inside the primary cell, we found that there is a specific form of epigenetic damage that are embedded inside the endoplasmic reticulum, which is a specific area of the cell in the nuclear membrane. Sorry, and not in the nuclear membranes. It's a different place. And so normal trauma techniques are not really efficient on these epigenetic damages because it's not the same way to access them. So how we do something about it? Well, we designed the body association technique based on the understanding of how the primary cell works to solve this, and it works. And this allowed us to have, nowadays we have pay for results policy when we do trauma healing, because we know how trauma works. So we know that we have the techniques that can deal with different issues inside the primary cell. Is it a basic trauma, or is it a trauma with an association, which means it's linked to survival and the body will fight trauma healing? Therefore, we know that we have to use the body association technique, and so on and so on. And this is amazing, because this could only be done if you have an understanding of what's going on, if you have a model. And if we didn't have this model of the primary cell, we never could have created this body association technique, unless we've been very, very lucky. But would have we been, sorry for my English, would have we been able to recognize what we were doing? That's not sure. So having an underlying models that you understand is really, really, really helpful. Another application is schizophrenic voices. And we've observed that they are caused by a fungal infection. And this fungal infection is transmitted from parent to child before birth because of epigenetic damages. And so we wonder, okay, what if we repair those epigenetic damages? And we can do it because, remember, epigenetic damages are trauma. So if we use trauma healing on this moment, what does it do? In theory, it should confer immunity against the fungus, and those voices should disappear. And actually, it works. And we created a technique called the silent mind technique. And if you want to know all about it in details, I encourage you to look into this book written by Dr. Grant McFetridge, Silence the Voices. You can find it in your library or online. And it explains all of the scientific process of discovery, from very early observations to trial and error until these techniques were designed. It's a fascinating read. All right, so how do we use this to find the origin of diseases of unknown etiology with a scientific approach? Well, we use a scientific approach. So we ask ourselves, okay, this disease, can we track it inside the primary cell? And if we can, then, okay, can we use regression to identify when the problem starts in development? And if we do, okay, let's try to heal the epigenetic damages. Can we heal traumas and repair this event? And if it works, then would healing those trauma change the biology of the disease? That's basically, really basically, how we do it. It's much more complex and nuanced, but basically we design those processes based on these questions, and we try to verify, does it work? Do we have an understanding of the biology which is thorough enough, good enough to have the effects that we want to have? And we're always testing and finding new processes and new ways of doing those processes, because our methodology and our technology has changed over time a lot. But this is basically the thinking patterns that we use. So I'm going to end this presentation here. So I explained to you what is psychobiology. I didn't talk about psychoimmunology. So a short word on it, because you're going to hear about it all throughout the symposium. But we've observed that pre-birth infections are possible. You can be infected by a bacteria or a fungus or a virus before you're born. Why? Because your parent carries it. And there are moments in development where there is possibility for pathogens to infect us. And we've seen it again and again. And we've also practiced healing the traumas that happened at those moments. And the pathogen disappeared from the primary cell. I just mentioned it before with the Salman technique. It's exactly that, but we've done it with all the pathogens too. So psychoimmunology is finding how can we change our immune system so that it can see that pathogens are not good. And one of the hypotheses that we have is that those pre-birth infections so-called open the door for infections later on in life, because pathogens use similar chemical signals. So to explain that, if we've been infected pre-birth by, let's say, bacteria, the bacteria will use a specific chemical signals, and because we have epigenetic damages, we are sensible to it, so we let the bacteria in. Now, later on in life, if we meet another bacteria or another pathogen which is using the same chemical signals, we are much more prone to be sensitive, to be vulnerable to it. Actually, if you go back to last year's symposium and the symposium of 2 years ago, you will have examples of that. So psychobiology is a branch of psychobiology, and it's a science of designing epigenetic repair processes that makes us immune to certain pathogens, viruses, bacteria, fungi, and so on. So here are a few observable phenomena that I wanted to mention to conclude this presentation today, because I just wanted to help you see the potential of this work. And this work can help us find potential treatments for mental disorders or physical disorders that are thought to be incurable, such as autism or type 1 diabetes, and also phenomena that are supposed to be impossible to even comprehend. A good example are the meridians from Chinese medicine. So the Western science haven't found those meridians and doesn't really know where they are and why they exist. However, Western science also have thousands of clinical studies on acupuncture which demonstrate an effect, and a powerful effect. And it's hard for generally for biologists and doctors in the Western world to reconcile the efficiency that we see again and again and again with acupuncture with this model of meridians, because they cannot see the meridians. Well, it turns out we have the solution, because meridians can be seen inside the primary cell. Therefore, acupuncture interacts with those meridians and have a biological effect inside the primary cell. And if there is a change in the primary cell, as you understood, there is a change in the whole body. Hence, that's how Chinese medicine works, at least the acupuncture part of it. In the next presentation, you will hear about other topics that we work on, such as OCD and also pathogens and psychoimmunology. There's so much to say about it that I'm going to leave you with my good colleague, Dr. Mary Pellicer. And